Progress just got simpler. BLI is an optical technique that analyzes the interference pattern of white light reflected from two surfaces, a layer of immobilized protein on a biosensor tip and an internal reference layer. Any change in the number of molecules bound to the biosensor tip causes a shift in the interference pattern, which is recorded in real time. A proprietary protein coating at the biosensor tip acts as a binding partner, providing reproducible coupling of target molecules while minimizing non-specific binding. The measurement is plate-based. An aqueous buffer is used to generate a baseline before the sensor dips into a sample containing the analyte. The analyte then binds to the sensor surface, and the binding interaction is followed in real time to reveal the association kinetics. The sensor tip is then dipped into fresh buffer, and the dissociation can be monitored until the regeneration of the biosensor's surface. This real-time analysis provides precise and accurate data on binding specificities, analyte concentrations, and rates of association and dissociation. A unique characteristic of BLI is that only analyte binding or dissociation can shift the interference pattern and generate a response profile. Unbound molecules, changes in the refractive index or pH, or other matrix effects do not affect results, making this technology well-suited to measurements in crude samples. In addition, BLI is easy to use, making assay development and optimization a much faster process for a shorter time to results. This fluidic-free approach eliminates sample loading or cleaning steps between measurements.
Today we are going to study interaction between CD70 and CD27 using BLI. Here are the experimental parameters we have. We are going to immobilize biotinylated CD72 antigen on the stropavidin biosensor at 50 nanomolar concentration. The ligand concentration range which we are going to use is from 0 to 5 micromolar. The buffer system which we have has BSA and twin to avoid non-specific binding. Here in the blocking buffer we have PBS buffer with BSA twin 20 and biotin. We have made the ligand in the blocking buffer as it will occupy the unbound surface on the stropavidin biosensor tips. We have reference biosensor and we have 0 ligand concentration as controls for the blank subtraction. The instrument which we are using today which is Octet 384 which is designed to work with 384 well plates and has 16 channels. Octet 384 can measure 12 dissociation constants at a time in a high throughput manner. Next we will talk about setting a BLI experiment in the Discovery software tool. The first step is to assign a sample to each well in the plate definition. We don't need to assign the reference now as we can do that in the ladder analysis. So we are assigning these wells in 384 plate to the samples and we are using 16 channel sensors. Next step we go to the assay definition where we set up the assay with baseline loading baseline 2 association dissociation steps. Baseline will ensure that the tips are well equilibrated in the buffer. Loading is very crucial step here as we want optimum amount of target protein for the immobilization. There should not be oversaturation of target on the sensor as there can be protein precipitation on the tip or there can be steric hindrances for the protein-protein interaction, and we should set this for all the sensor to maintain homogeneity in the assay. Next step is baseline 2 where we will remove any unbound biotinylated target. Then association and dissociation. At least 10 minutes is necessary for these steps. Then we go to the sensor arrangement, where we will assign each well to a stropavidin biosensor. Next click on Review Experiment. This is a dry run of the experiment. As you click on the arrow, you can see the sensor will move from the wells and it goes from baseline loading baseline to association and dissociation. Now go ahead and run the experiment. Oh wow, this is amazing! You can see real-time analysis of the protein-protein interaction. So this is a baseline step. All the 16 biosensors have different colors and there is a very less response as this is just buffer which is blank. There is no bound target antigen. You can see loading where we have got maximum around 0.5 of the loading which is optimum loading. We can see concentration-dependent association of the ligand with target. Now we need to analyze this data for measuring kinetic parameters such as dissociation constant. Next, we will perform the data analysis for the measuring kinetic parameters of CD70 and CD27 interaction. Here we have to assign the reference sensor from the wells for the blank subtraction. Then we go to reference sample, and in each well now we have different concentration ranges of the ligand, and we need to assign the concentrations used in the assay. We have used CD27 from 0 to 5 micromolar concentration in 2-fold dilutions. Now align y-axis the average of baseline step. Then you go to kinetic analysis, and then we are analyzing both association and dissociation, both the steps here for the kinetic parameters measurement. Then hit apply and calculate response. On the right bottom side you can see the graph of response versus ligand concentration, and here you will get a dissociation constant and other parameters for protein-protein interaction. All these analysis report from this software you can export into Excel file. This is how we do the KD analysis using BLI.